Chapter 14, Respiration in Plants. All of us breathe to live, but why is breathing so essential to life? What happens when we breathe? Also, do all living organisms, including plants and microbes, breathe? If so, how? All living organisms need energy for carrying out daily life activities, be it absorption, transport, movement, reproduction, or even breathing. Where does all this energy come from? We know we eat food for energy, but how is this energy taken from food? How is this energy utilized? Do all foods give the same amount of energy? Do plants eat? Where do plants get their energy from? And microorganisms, for their energy requirements, do they eat food? You may wonder at the several questions raised above. They may seem to be very disconnected, but in reality, the process of breathing is very much connected to the process of release of energy from food. Let us try and understand how this happens. All the energy required for life processes is obtained by oxidation of some macromolecules that we call food. Only green plants and cyanobacteria can prepare their own food. By the process of photosynthesis, they trap light energy and convert it into chemical energy. This is stored in the bonds of carbohydrates like glucose, sucrose, and starch. We must remember that in green plants too, not all cells, tissues, and organs photosynthesize. Only cells containing chloroplast that are most often located in the superficial layers carry out photosynthesis. Hence, even in green plants, all other organs, tissues, and cells that are non green need food for oxidation. Hence, food has to be translocated to all non green parts. Animals are heterotrophic, that is, they obtain food from plants directly, herbivores, or indirectly, carnivores. Saprophytes like fungi are dependent on dead and decaying matter. What is important to recognize is that ultimately all the food that is required for life processes come from photosynthesis. This chapter deals with cellular respiration, or the mechanism of breakdown of food materials within the cells to release energy, and the trapping of this energy from for synthesis of ATP. Photosynthesis, of course, takes place within the chloroplast in the eukaryotes, whereas the breakdown of complex molecules to yield energy takes place in the cytoplasm and in the mitochondria, also only in eukaryotes. The breaking of C-C bonds of complex compounds through oxidation within the cell, leading to release of considerable amount of energy, is called respiration. The compounds are oxidized during this process are known as respiratory substrates. Usually, carbohydrates are oxidized to release energy, but proteins, fats, and even organic acid can be used as respiratory substrate in some plants under certain conditions. During oxidation within a cell, all the energy contained in respiratory substrate is not released free into the cell or in a single step. It is released in a series of slow stepwise reactions controlled by enzymes, and it is trapped as chemical energy in a form of ATP. Hence, it is important to understand that the energy released by oxidation in respiration is not, or rather cannot be used directly, but it is used to synthesize ATP, which is broken down whenever and wherever energy need to be utilized. Hence, ATP acts as the energy currency of the cell. This energy trapped in the ATP is utilized in various energy requiring processes of the organism, and the carbon skeleton produced during respiration is used as precursors for biosynthesis of other molecules in the cell. 14.1, do plants breathe? Well, the answer to this question is not quite so direct. Yes, plants require O2 for respiration to occur, and they also give out CO2. Hence, plants have system in place that ensures the availability of O2. Plants, unlike animals, have no specialized organ for gaseous exchange, but they have stomata and lenticels for this purpose. There are several reasons why plants can get along without respiratory organs. First, each plant part takes care of its own gas exchange needs. There is very little transport of gases from one plant part to the other. Second, plants do not present great demands for gas exchange. Roots, stems, and leaves respire at a rate far lower than animals do. Only during photosynthesis, a large volume of gases exchange, and each leaf is well adapted to take care of its own need during these periods. When cell photosynthesize, availability of O2 is not a problem in these cells since O2 is released within the cell. Third, the distance that gases must diffuse even in large bulky plant is not great. Each living cell in the plant is located quite close to the surface of the plant. This is true for leaves. You may ask, but what about thick woody stem and roots? In stem, the living cells are organized in thin layer inside and beneath the bark. They also have openings called lenticels. The cells in the interior are dead and provide only mechanical support. Thus, most cells of the plant have at least a part of their surface in contact with air. This is also facilitated by the loose packing of parenchyma cells in leaves, stems, and roots, which provide an interconnected network of air spaces. The complete combustion of glucose, which produces CO2 and H2O as end product, yield energy, most of which is given out as heat. C6H12O6 plus 6O2 gives 6CO2 plus 6H2O plus energy. If this energy is to be useful to the cell, it should be able to utilize it to synthesize other molecules that the cell requires. The strategy that the plant cell uses is to catabolize the glucose molecule in such a way that not all the liberated energy goes out as heat. The key is to oxidize glucose not in one step but in several small steps, enabling some steps to be just large enough such that the energy release can be coupled to ATP synthesis. How this is done? Essentially, the story of respiration. During the process of respiration, oxygen is utilized and carbon dioxide and water and energy are released as products. The combustion reaction requires oxygen, but some cells live where oxygen may or may not be available. Can you think of any of such situation or organism where O2 is not available? There are sufficient reasons to believe that first cell on this planet lived in an atmosphere that lacked oxygen. Even among present-day living organisms, we know of several that are adapted to anaerobic conditions. Some of these organisms are facultative anaerobes, while in others the requirement of anaerobic condition is obligate. In any case, all living organisms retain the enzymatic machinery to partially oxidize glucose without the help of oxygen. This breakdown of glucose to pyruvic acid is called glycolysis. 14.2 Glycolysis. The term glycolysis has originated from the Greek word glykos for sugar and lysis for splitting. The scheme of glycolysis was given by Gustav Embden, Otto Meyerhoff, and J. Parnas, and is often referred to as EMP pathway. In anaerobic organism, it is the only process in respiration. Glycolysis occurs in the cytoplasm of the cell and is present in all living organisms. In this process, glucose undergo partial oxidation to form two molecules of pyruvic acid. In plants, this glucose is derived from sucrose, which is the end product of photosynthesis, or from storage carbohydrates. Sucrose is converted into glucose and fructose by the enzyme invertase, and these two monosaccharides readily enter the glycolytic pathway. Glucose and fructose are phosphorylated to give rise to glucose 6 phosphate by the activity of the enzyme hexokinase. This phosphorylated form of glucose then isomerizes to produce fructose 6 phosphate. Subsequent steps of metabolism of glucose and fructose are same. The various steps of glycolysis are depicted in figure 14.1. In glycolysis, a chain of 10 reactions under the control of different enzymes takes place to produce pyruvate from glucose. While studying the steps of glycolysis, please note the steps at which utilization or synthesis of ATP, or in this case NADH plus H plus, takes place. ATP is utilized at two steps. First, in the conversion of glucose into glucose 6 phosphate, and second in the conversion of fructose 6 phosphate to fructose 1,6 bisphosphate. The fructose 1,6-bisphosphate is split into dihydroxyacetone phosphate and 3-phosphoglyceraldehyde PGAL. We find that there is one step where NADH plus H plus is formed from NAD plus. This is when 3-phosphoglyceraldehyde PGAL is converted to 1,3-bisphosphoglycerate BPGA. Two redox equivalents are removed in the form of two hydrogen atoms from PGAL and transferred to the molecule of NAD+. PGAL is oxidized and with inorganic phosphate to get converted into BPGA. The conversion of BPGA to 3-phosphoglyceric acid PGA is also an energy yielding process. This energy is trapped by the formation of ATP. Another ATP is synthesized during the conversion of PEP to pyruvic acid. Can you then calculate how many ATP molecules are directly synthesized in this pathway from one glucose molecule? Pyruvic acid is the key product of glycolysis. What is the metabolic fate of pyruvate? This depends on the cellular need. There are three major ways in which different cells handle pyruvic acid produced by glycolysis. These are lactic acid fermentation, alcoholic fermentation, and aerobic respiration. Fermentation takes place under anaerobic condition in many prokaryotes and unicellular eukaryotes. For the complete oxidation of glucose to CO2 and H2O, however, organisms adopt Krebs cycle, which is also called as aerobic respiration. This requires O2 supply. 14.3 Fermentation. In fermentation, say by yeast, the incomplete oxidation of glucose is achieved under anaerobic condition by set of reactions where pyruvic acid is converted to CO2 and ethanol. The enzyme pyruvic acid decarboxylase and alcohol dehydrogenase catalyze these reactions. Other organisms like some bacteria produce lactic acid from pyruvic acid. The steps involved are shown in figure 14.2. In animal cells also, muscles during exercise when oxygen is inadequate for cellular respiration, pyruvic acid is reduced to lactic acid by lactate dehydrogenase. The reducing agent is NADH plus H plus, which is reoxidized to NAD plus in both the processes. In both lactic acid and alcoholic fermentation, not much energy is released. Less than 7% of the energy in glucose is released and not all of it is trapped as high energy bonds of ATP. Also, the processes are hazardous. Either acid or alcohol is produced. What is the net ATPs that is synthesized? Calculate how many ATP are synthesized and deduct the number of ATP utilized during glycolysis. When one molecule of glucose is fermented to alcohol and lactic acid, yeast poison themselves to death glucose when the concentration of alcohol reaches about 13%. What then would be the maximum concentration of alcohol in beverages that are naturally fermented? How do you think alcoholic beverages of alcohol content greater than this concentration are obtained? What then is the process by which organisms can carry out complete oxidation of glucose and extract the energy stored to synthesize a large number of ATP molecule needed for cellular metabolism? In eukaryotes, these steps take place within the mitochondria and this require O2. Aerobic respiration is a process that leads to the complete oxidation of organic substance in the presence of oxygen and releases CO2, water and large amount of energy present in the substrate. This type of respiration is most common in higher organisms. We will look at these processes in the next section. 14.4 Aerobic respiration. For aerobic respiration to take place within the mitochondria, the final product of glycolysis, pyruvate, is transported from the cytoplasm into the mitochondria. The crucial events in aerobic respiration are the complete oxidation of pyruvate by the stepwise removal of all the hydrogen atoms, leaving three molecules of CO2. The, the passing on of the electrons removed as a part of hydrogen atom to molecular oxygen with simultaneous synthesis of ATP. What is interesting to note is that the first step takes place in the matrix of the mitochondria while the second process is located on the inner membrane of the mitochondria. Pyruvate, which is formed by the glycolytic catabolism of carbohydrate in the cytosol after it enters the mitochondrial matrix, undergo oxidative decarboxylation by a complex set of reactions catalyzed by pyruvic dehydrogenase. The reactions catalyzed by pyruvic dehydrogenase require the participation of several coenzymes, including NAD plus and coenzymes. Pyruvic acid plus coenzyme A plus NAD plus gives, in the presence of Mg2 plus and pyruvate dehydrogenase, gives acetyl coenzyme A plus CO2 plus NADH plus H plus. During this process, two molecules of NADH are produced from the metabolism of two molecules of pyruvic acid produced from one glucose molecule during glycolysis. The acetyl coenzyme A then enters a cyclic pathway, tricarboxylic acid cycle, or more commonly called as Krebs cycle, after the scientist Hans Krebs, who first elucidated it. 14.4.1 Tricarboxylic acid cycle. The TCA cycle starts with the condensation of acetyl group with oxalic acid, oxaloacetic acid, and water to yield citric acid. Figure 14.3. The reaction is catalyzed by the enzyme citrate synthase and a molecule of CoA is released. Citrate is then isomerizes to isocitrate. It is followed by two successive steps of decarboxylation leading to the formation of alpha ketogutaric acid and then succinyl coenzyme A. In the remaining steps of citric acid cycle, succinyl coenzyme A is oxidized to oxaloacetic acid, allowing the cycle to continue. During the conversion of succinyl coenzyme A to succinic acid, a molecule of GTP is synthesized. This is the substrate level phosphorylation. A couple of reaction GTP is in a couple of reaction GTP is converted to GDP with the simultaneous synthesis of ATP from ADP. Also there are 3 points in the cycle where NAD plus are reduced to NADH plus H plus and 1 point where FAD plus is reduced to FADH2. The continued oxidation of acetyl coenzyme A via the TCA cycle requires the continued replenishment of oxaloacetic acid, the first member of the cycle. In addition, it also requires the regeneration of NAD plus and FAD plus from NADH and FADH2 respectively. The summary equation for this phase of respiration may be written as follows: pyruvic acid plus 4 NAD plus FAD plus 2H2O plus ADP plus inorganic phosphate in mitochondrial matrix give 3CO2 plus 4NADH plus 4H plus FADH2 plus ATP. We have till now seen that glucose has been broken down to release CO2 and 8 molecules of NADH plus H plus. 2 of FADH2 have been synthesized beside just 2 molecules of ATP in TCA cycle. You may be wondering why we have been discussing respiration at all. Neither O2 has come into this picture nor the promised large number of ATP has yet been synthesized. Also, what is the role of NADH plus H plus and FADH2 that is synthesized? Let us now understand the role of O2 in respiration and how ATP is synthesized. 14.4.2 Electron Transport System ETS and Oxidative Phosphorylation. The following steps in the respiratory process are to release and utilize the energy stored in NADH plus H plus and FADH2. This is accomplished when they are oxidized through the electron transport system and the electrons are passed on to O2, resulting in the formation of H2O. The metabolic pathway through which the electron passes from one carrier to another is called the electron transport system ETS. Figure 14.4, and it is present in the inner mitochondrial membrane. Electrons from NADH produced in the mitochondrial matrix during citric acid cycle are oxidized by an NADH dehydrogenase complex 1 and electrons are then transferred to ubiquinone located within the inner membrane. Ubiquinone also receives reducing equivalents via FADH2 complex 2 that is generated during oxidation of succinate in the citric acid cycle. The reduced ubiquinol is then oxidized with the transfer of electron to cytochrome C via cytochrome Bc1 complex. Complex 3. Cytochrome C is a small protein attached to the outer surface of the inner membrane and act as a mobile carrier for transfer of electron between complex 3 and 4. Complex 4 refers to cytochrome C oxidase complex containing cytochromes A and A3 and two copper centers. When the electron passes from one carrier to another via complex 1 to 4 in the electron transport chain, they are coupled to ATP synthase complex 5 for the production of ATP from ADP and inorganic phosphate. The number of ATP molecules synthesized depend on the nature of the electron donor. Oxidation of one molecule of NADH gives rise to 3 molecule of ADP, 3 molecule of ATP, while that of one molecule of FADH2 produce 2 molecule of ATP. Although the aerobic process of respiration takes place only in the presence of oxygen, the role of oxygen is limited to the terminal stage of the process. Yet the presence of oxygen is vital since it drives the whole process by removing hydrogen from the system. Oxygen acts as the final hydrogen acceptor, unlike photophosphorylation, where it is the light energy that is utilized for the production of proton gradient required for phosphorylation. In respiration, it is the energy of oxidation reduction utilized for the same. It is for this reason that the process is the oxidative phosphorylation. You have already studied about the mechanism of membrane linked ATP synthesis as explained by chemoosmotic hypothesis in the earlier chapter. As mentioned earlier, the energy released during the electron transport system is utilized in synthesizing ATP with the help of ATP synthase complex 5. The complex consists of two major components F1 and F0. Figure 14.5. In F1, headpiece is a peripheral membrane protein complex and contain the site for synthesis of ATP from ADP and inorganic phosphate. F naught is an integral membrane protein complex that forms the channel through which protons cross the inner membrane. The passage of protons through the channel is coupled to the catalytic site of the F1 component for the production of ATP. For each ATP produced, 2H plus passes through F0 from the intermembrane space to the matrix down the electrochemical proton gradient. 14.5 The respiratory balance sheet. It is possible to make calculation of the net gain of ATP for every glucose molecule oxidized, but in reality, this can remain only a theoretical exercise. These calculations can be made only on certain assumptions that there is a sequential, orderly pathway functioning with one substrate forming the next with glycolysis, TCA cycle, and ETS pathway following one after other. The NADH synthesized in glycolysis is transferred into mitochondria and undergo oxidative phosphorylation. None of the intermediates in the pathway are utilized to synthesize any other compound. Only glucose is being respired, no other alternative substrate are entering in the pathway at any of the intermediary stages. But this kind of assumptions are not really valid in living system. All pathways work simultaneously and do not take place one after other. Substrates enter the pathways and are withdrawn from it as and when necessary. ATP is utilized as and when needed. Enzymatic rates are controlled by multiple means, yet it is useful to do this exercise to appreciate the beauty and efficiency of the living system in extraction and storing energy. Hence, this Hence, there can be a net gain of 38 ATP molecules during aerobic respiration of one molecule of glucose. Now let us compare fermentation and aerobic respiration. Fermentation accounts for only a partial breakdown of glucose, whereas in aerobic respiration it is completely degraded to CO2 and H2O. In fermentation, there is net gain of only two molecules of ATP for each molecule of glucose degraded to pyruvic acid, whereas many more molecules of ATP are generated under aerobic conditions. NADH is oxidized to NAD plus rather slowly in fermentation. However, the reaction is very vigorous in case of aerobic respiration. 14.6 Amphibolic Pathway. Glucose is the favored substrate for respiration. All carbohydrates are usually first converted into glucose before they are used for respiration. Other substrate can also be respired as has been mentioned earlier, but then they do not enter the respiratory pathway at the first step. See figure 14.6 to see the points of entry of different substrate in the respiratory pathway. Fats would need to be broken down into glycerol and fatty acid first. If fatty acids were to be respired, then they would first be degraded to acetyl coenzyme A and then enter the pathway. Glycerol would enter the pathway after being converted to PGAL. The proteins would be degraded by the protease, and the individual amino acid after deamination, depending on their structure, would enter the pathway at some stage within the Krebs cycles or even as pyruvate or acetyl coenzyme A. Since respiration involves breakdown of substrate, the respiratory process has traditionally been considered a catabolic process and the respiratory pathway as a catabolic process, but is this understanding correct? We have discussed all above at which points in the respiratory pathways different substrate would enter if they were to be respired and used to derive energy. What is important to recognize is that it is these very compounds that would be withdrawn from the respiratory pathway for the synthesis of the said substrate. Hence, fatty acid would be broken down to acetyl coenzyme A before entering the respiratory pathway when it is needed as a substrate. But when the organism need to synthesize fatty acid, acetyl coenzyme A would be withdrawn from the respiratory pathway for it. Hence, the respiratory pathway come into the picture both during breakdown and synthesis of fatty acid. Similarly, during breakdown and synthesis of proteins to respiratory intermediates from the link forms the link. Breaking down processes within the living organism is catabolism and synthesis is anabolism. Because the respiratory pathway is involved in both anabolism and catabolism, it would hence be better to consider the respiratory pathway as an amphibolic pathway rather than as a catabolic one. 14.7 Respiratory Quotient. Let us now look at another aspect of respiration. As you know, during aerobic respiration, O2 is consumed and CO2 is released. The ratio of the volume of CO2 evolved into the volume of O2 consumed in respiration is called the respiratory quotient, RQ, or respiratory ratio. RQ is equal to volume of CO2 evolved upon volume of O2 consumed. The respiratory quotient depends upon the type of respiratory substrate used during respiration. When carbohydrates are used as substrate and are completely oxidized, the RQ will be 1 because equal amount of CO2 and O2 are evolved and consumed respectively as shown in the equation below. C6H12O6 plus 6O2 gives 6CO2 plus 6H2O plus energy. RQ is equal to 6CO2 upon 6O2 is equal to 1. When fats are used in respiration, the RQ is less than 1. Calculation for a fatty acid tripalmitine if used as a substrate is shown. 2 molecule of C51H98O6 plus 145O2 gives 102 CO2 plus 98 H2O plus energy. RQ is equal to 102 CO2 upon 145 O2, which is equal to zero point 0.7. When proteins are respiratory substrate, the ratio would be about 0.9. What is important to recognize is that in living organisms, respiratory substrate are often more than one, but pure proteins or fats are never used as respiratory substrates.